Hi everyone. I'm sorry for the delay in posting this lecture. We've had a lot of other mini lectures that have been needed to be posted before then, so we weren't able to do this one. Also, since I've been sick and getting chemotherapy and stuff, it's been difficult to get the energy to do it. But here we are. This is the 10th lecture in the genetics series. This is going to be about linkage and mapping. Fun stuff of genetics. It's going to cover chapter 7 in the online Open Genetics Nickel and Barrett book, and then in the other text that I have available for you through Open Genetics, it's in chapter 10 of that one. You can find the links to that in the information section below. So what we're going to cover today is linkage, recombination, how linkage reduces recombination frequency, and then we're going to talk about how linkage is detected, what exactly is happening there, how the crossover frequencies change when there are linked loci available, and then we're going to talk about how we can calculate distances between different genes in map units, inferring through those recombination data that we get from those crosses using 3-point crosses specifically. So what exactly is linkage? Let's start with that. So we've talked about how, one of the main principles of Mendelian genetics is that each gene is going to be independently assorted, right? As with the Mendel's second law. However, when they are genes on the same chromosomes, especially those that are really, really close together, you can imagine if you have two small genes or you have genes that are very, very close to each other, there is a likelihood that they can get swapped over kind of together. And that phenomena is what is called linkage. When you have that exception to the second law of independent assortment, where genes that are on the same arm of the chromosome or that are on the same chromosome are more likely to be crossed over together, leading to recombination frequencies that don't necessarily follow the second law of independent assortment and Mendelian genetics. It is very unlikely that genes are going to cross over when they are on different sides of the chromosomal arm. That usually doesn't happen. But when there are genes that are going to be in the same, right next to each other, they're very possibly linked and they can go along together. So here is an example where if you're looking at, for example, genes that are F and C, they're far apart, and there's very likely that you will have a combination happening in between those places, and that's going to lead to just the normal Mendelian ratios. However, when genes are very, very close to each other, right next to each other, they become a little bit more linked, and there's much more unlikely scenario that there's a crossover happening between them. So the chromosomal theory of inheritance, which is, the way that we look at Mendelian genetics, the way we look at genes, it's established that genes in each chromosomes are usually present linearly. There are many, many genes present on each arm of the chromosome in either direction. All these genes that are located on the same chromosome are obviously going to move to the same pole during cell division, right? They're not going to be parts of that arm going. The whole arm is going to go. The whole chromosome is going to move. So as a consequence, obviously, that those genes are not going to show independent assortment perfectly the way you normally see with independent genes. And they would be more likely to be inherited together. And then within the chromosome, the closer they are, the more likely that they will be linked and moved together versus having recombination events between them due to crossover. So there are 3 different types of linkage that can happen, right? And some is more involved than others. So one is simply how likely is it that there's going to be a crossover happening between two genes that are present on that chromosome. So individual chromosomes, if genes are very far apart, they can still be independently assorting because they're far enough apart that there'll be enough crossover events happening that you end up with still Mendelian ratios at the end of the day. However, if they're very close to each other, less likelihood of crossing over happening, obviously less likelihood that you're going to get those independent ratios. Then what genes are involved, right? Where they are they on different chromosomes? Are they on the same chromosomes? Are they somehow regulated by the same thing maybe? Those type of things. And then finally, chromosomal involvement. Are they on the same chromosome versus are they on different chromosomes? Genes on the same chromosome are more likely to be linked than genes on separate chromosome, obviously, because they're not linked through that mechanism. So based on crossing over, there are two different things that you can have. We're going to talk about each one of those, 3 scenarios and look at them a little bit more in detail. So just looking at crossing over, just because they are there on the chromosome, same chromosome, does not mean that there's going to be complete linkage or complete linkage in between them. So there are cases where you would have complete linkage of genes. That means that those genes that you're talking about are always going to be together. An example of that, there's no recombination. 0 crossover, 0 recombination. Well, in typical diploid system, you think that's not possible, but there are many such scenarios already available in our environment. Males in Drosophila do not undergo recombination, so their genes are completely linked. They're always going to be the same on that chromosome arm that is going to be coming from the males in the Drosophila. Similarly, females of silkworm have the same thing. They do not have recombination. So if there's complete absence of recombination or crossing over, then those genes are completely linked if they're on the same chromosome or they're in that organism of that particular sex or that particular type. Incomplete or partial linkage happens when there is still some crossing over happening. That's our typical type of linkage that you see with crossing over. So in typical cases, in any organism that has recombination, genes on the same chromosome, especially on the same arm of the chromosome, are going to have incomplete or partial linkage. So there will be some frequency of crossing over happening. It's always likely some percentage, even if it's 0.0001, there is some likelihood that 2 genes that are even right next to each other may have a crossover possibility. And so those will not be 100% linked. They're not always going to be together. They will be what we call incomplete or partial linkage. So when they're linked, obviously there's going to be less variability because there's less crossover happening between those areas that we are talking about. So if the 2 loci are completely linked, those alleles are always going to be the same. The only possibility you're going to get is the parental ones that you're getting, right? That's a complete linkage. However, if there is some kind of crossover happening, right, then you can have all the different phenotypes available, all the different possibilities available where there is recombination happening and you are getting different combinations. However, those percentages are not going to match Mendelian genetics percentages. Looking at the second scenario, which is looking at genes involved, this depends on whether you have all dominant genotype or all recessive alleles that are linked together. Sometimes you have, and there are 2 different things that we look at in here. There's a coupling phase and a repulsion phase. So in some cases, the dominant forms or the recessive allele, either dominant or the recessive alleles are linked together. And in those cases, those are called coupled genes or coupled alleles. So you will have the dominant or the recessive alleles present on the same chromosome, right? So you'll only have the dominant ones on the chromosome, or you'll only have the recessive allele present on the chromosome for a particular 2 genes that you're looking at. And in those cases, those are somehow coupled in there. And in repulsion phase, you have dominant alleles of some genes that are linked with recessive alleles of other genes. So whenever you have a dominant allele of gene A, you have a recessive allele of gene B, for example. And in that case, that's what we call a repulsion phase. So in those cases, they are linked, but they're linked in a very specific way. They're not linked in a typical linkage way where it's still a sorting and you can still have multiple combinations. In this case, if those genes are coupled or repulsed, they will only have both dominant or both recessive if they're coupled, or they will have the opposing ones if they are repulsed. So here is an example. Okay, so when we're looking at any scenario like this, we want to know which one are our recombinants and which one are our parental genotypes that in the gametes that we are getting. So here are two examples where in the first example you have both, in both cases these are two genes, and in the first example are the two parents. They have one dominant gene and one recessive gene. And the second one, they have all homozygous dominant or homozygous recessive. And in this case, when they combine together and you get your gametes, you get your cell that's going to produce the gametes, you'll get two gametes that are going to be your parentals, which are going to have, again, the exact same gametes that the original parent was able to produce, right? So this was one dominant and one recessive, same as the parents. However, it's going to also now, with crossing over, be able to produce both dominant or both recessive gametes. And those two that are going to give you those possibilities, the crossed over ones, are your recombinants, and the original gametes, like the ones of the parents, are the parentals. In this case, again, these are going to have different gametes as your parent or the recombinant, because you're starting off with different things. So in this particular parent, because it was homozygous recessive, the only gamete you were going to get was homozygous recessive for both genes. And then once it's combined with that second parent, the F1 progeny, you have two heterozygous genes, and so you can have all four possibilities. Now, on the all four possibilities of the gametes, the ones that are going to be your parental are homozygous recessive or homozygous dominant. Those are your same as the original parent ones. And then the other two are going to be your crossover, which are your recombinants. So you should be able to identify which one of the gametes are recombinants and which genotypes are recombinants and which genotypes are parental. So here we look at our an actual example of an experiment to measure recombination frequency between two loci. So we're just looking at two different genes, and we're trying to see if it matches Mendelian genetics or not in this case. So two genes at the same time. And here in your parents, you have a heterozygous, two heterozygous parents, where they're in opposing manner, right? So one is homozygous recessive for the first allele, and homozygous dominant for the second, or for first gene, and homozygous dominant for the second gene, and the other parent is homozygous dominant for the first gene and homozygous recessive for second. So in your F1, every progeny is going to be heterozygous for both alleles, or for both genes. And now when you look at F2, you are going to get different types of genotypes. And here, if you have your homozygous recessive here, and then heterozygous, remember that you're going to have some recombination happening to create that individual, right? And your original ones are still going to be, these are your parentals. So here we have all those possibilities, and the way we look at that is by looking at gametes from the dihybrid. So gametes from here, that were the possibilities. So the gamete possibilities that you had here were obviously big A, little b, right, which would be one of your parentals, little a, little b, which would be not a parental, that's recombinant, and then big B, little b, which is going to be one of the parentals, and then little b, little b. So here are your different possibility for the gametes. So you have far phenotype, whether it's brown or white, and tail phenotype, whether it's short or long. Our original parents had short tail and brown, or long and white. So that's how we look at that. So short and brown, that's one of our parents. So that's going to be our parental. It gives you the number of progeny that had that phenotype, and then the gametes from the dihybrid that would have gotten from that would have given you that. So this is little a and big B. That's one of the parental gametes, and that's what you would write here. Second is long and white, and so they tell you the number of progeny and again the gametes from the dihybrid, and that's again one of your parentals, right? Because white and heterozygous is that. And then the other two combinations are going to be your recombinants because they weren't one of the parents. So short and white or long and brown. Those are different than the originals, so they are going to be your recombinants. So your recombination frequency is going to be to add up the number of progeny you have with recombinant phenotypes and divide that by total number of gametes that you have in all, which is all of the different possibilities. And so when we do that, we get 25%. Now remember, in an independent assortment, you would expect to see 50%, right? Equal amount. Here, it's not, which tells you that those genes are somehow linked, because they're giving you ratios different than the Mendelian genes. So based on the location of your chromosome, on the chromosome, linkage can be categorized into autosomal linkage. These are genes that are on the autosomes, not on the sex chromosomes X or Y, or allosomal or sex linkage, which is linkage of genes which are on the gene on X or Y chromosome. Because remember, the Y chromosome, everyone's going to be linked because that's the only one there. There's nobody to recombine with in, for example, humans. So one of the things we do when we look at linkage is we have to characterize the type of linkage that is happening. In our scenario, we look at whether it involves two or more genes that are located in the same chromosome. Are they on the same arm? Are they on a same arm, same place? So linear, are they present in linear fashion on there? Once that is there, then we know that it's going to reduce variability. And then we need to look at, do they involve either dominant or recessive allele? So are they coupled? Or do you have some dominant and some recessive, which is the repulsion phase? Then usually genes that are located close to each other are more likely to be linked and are going to have lower recombination frequency. Which is that strength. It depends on the distance between the linked genes. The more close to each other they are, the higher the strength of recombination frequency. So it will have lower recombination frequency as a result. And this can be determined from doing a test cross for progeny data, like the one that we just showed you, to calculate recombination frequencies. So to detect linkage, we do a test cross. This is the most common method, where we take our F' heterozygous at both loci, right, the way we just looked at, and we do a double recessive parent, and then we look at the progeny that you get as a result. So we have one heterozygous individual for both of the genes, both loci, and the other parent is going to be completely recessive. And then we look at the phenotypic ratio. If the phenotypic ratio of our progeny is 1 to 1 to 1 to 1 between all of them, parents and our recombinants, then it's going to follow Mendelian genetics and it's going to be no linkage. However, if your numbers are different than that 1 to 1 to 1 to 1 ratio, if they're not all equal, if it's not 50-50, then that indicates that there is a presence of some type of linkage. And how linked they are is going to be dependent on the recombination frequency based on all those factors we just talked about. So this is extremely important in plant breeding, because if we are trying to create a plant of a particular type, we want to limit the variability among the progeny to get exactly what it is that we want. So if you have two or more loci that are going to be controlling that trait that you are looking for, if they are linked, it's going to be easier to control them and simultaneously change them to meet our needs, versus if they are really, really far away. However, linkage is undesirable when your needed and unneeded genes are linked together, right? Because if my resistance gene is linked to a phenotype, a color that is not going to sell, then it's not really the best thing. So it depends how it is that we're looking at. But if they're advantageous genes that are linked, that can control the same phenotype that we are trying to protect or help or improve, then you can simultaneously remove it. Do that at the same time and make our job a whole lot easier. So what is different with crossing over versus linkage? Crossing over obviously leads to separation of these linked genes and can involve exchange of those segments between non-sister and sister chromatids of homologous chromosomes. You will have crossing over will never exceed 50%, so that's going to be our recombination frequency, and as a result it's going to increase variability by forming new gene combinations. Linkage, on the other hand, does the exact opposite. It keeps genes together. It involves individual chromosomes, right? Not between chromosomes. If they're on different chromosomes, they're already unlinked. In the same chromosome, however, it involves those individual chromosomes, and the number of linkage groups can never be more than haploid chromosome number, and it reduces variability. So next, we are going to talk about genetic mapping. Okay, so let's talk about how we can use this to map genes and to organize them on the chromosomes and learn more about their location. So by using this linkage method and looking at linked genes of chromosomes, we can create a genetic map, and it is a graphical representation of relative distance between any number of genes. These can be called gene map, chromosome map, or crossover map. The purpose and use of genetic mapping is essentially to determine the linear order and distance of separation among genes that are linked on the same chromosomes. That's how we did a lot of our sequencing once the sequence was available, how we mapped them out and display the exact location and arrangement and combinations of the different genes that are on each chromosome arm and where exactly they are and what they do. They're very useful, obviously, you can imagine, in predicting our crosses and predicting how we can change them and what can be changed and what cannot be changed in a particular organism based on the location of those genes. And it helps us, obviously, to understand complexity of the genetic organization in different species and also see similarities between them. It also portrays our underlying basis for the inherited traits in a given organism. This is great, extremely useful from evolutionary point because we can compare different species and help to see the different relationships that different organism and species have between each other based on what genes are present across all of them and how and their relative location. It also helps us find new information on how to diagnose and treat inherited human diseases, and also helps us find genes that are linked to diseases based on what comes up in different populations. It's very, very important in agriculture because it provides plants and animal breeders information on how they can improve their strains or their particular product in their selective breeding programs. So before we can start genetic mapping of any species, we need to know a few things, right? So the first thing we need to know is how many chromosomes are present in that species, and then total number of genes of that species, as well by undergoing hybridization experiments in between wild and mutant strains to try to see which ones are from just alleles of the same gene versus which ones are completely different genes to begin with. Once we have that information, then we know how many linkage groups we are looking at, right? So chromosomes are called linkage groups. Each chromosome is a linkage group, right? So, and they are going to contain the genes that are linked to each other. So if you have a gene on chromosome 4 and the other gene is on chromosome 9, then you already know they're not going to be linked. So in humans, you have 22 autosomal linkage groups and a allosomal sex linkage group, which is your X and Y. And X are 2 because it's an X chromosome and a Y chromosome. So there are a total of 24 linkage groups available in humans. Genes that are going to be very far apart on the same chromosomes are usually going to independently assort from each other and will end up being in different, almost with the same assortment as Mendelian genetics. Now, how do we measure? So, right, we always need a unit of measurement. So in mapping genetic distances. The arbitrary unit to measure intergene distance on the chromosome is morgans or centimorgans. It is map unit. One map unit is our arbitrary unit that we use typically, and then centimorgan is going to represent a distance over which 1% crossing over can be expressed. So a map unit is 1% of crossover, which means 1% recombination frequency. It represents linear distance on our chromosome, leading to 1% recombination frequency. And then Morgan units, it represents 100% crossover. So 1% crossover can be expressed as 1 centimorgan, because each one of them is going to be 1%. So to change recombination frequencies into centimorgans, we use map distances in the centimorgans. We basically call them 1-to-1. So for small distances, recombination frequency is less than 30%. That's what we essentially do. So if you have 1% recombination frequency, it's 1 centimorgan. 2%, it's 2. Two loci alone will not be able to tell you where they are exactly in the space of the chromosome, whether they are one is upstream of the other. You typically need 3 or more to be able to do that. So that's something to be mindful of. Also, there are other features that can impede recombination frequency, or that can change how those things are crossing over. For example, you have the telomeric sequences at the end, you have the centromeres in the center, right? So those are going to also be changing where recombination happens, and those values are going to be different, and those need to be taken into account as well. Some parts of chromosomes are typically your hotspots in each of the chromosome, and have way higher rate of recombination compared to others, and that can obviously change the recombination frequencies in there, and that can hinder your ability to calculate their map distances correctly. Other regions that are usually the heterochromatin will have reduced crossing over and will largely remain unchanged in some cases. So here is an example of some of those kind of gene maps that we may make. So what we would notice is that synthetic genes are genes that lie on the same chromosomes. So you have some examples of synthetic genes and how they are changing and how they are recombinant, where they are, their locations relative to each other in map distances, right? So today we use synteny as conservation of blocks of order with 2 sets of chromosomes that are being compared with each other. So looking at 2 different chromosomes and looking at how the recombination frequencies are changing between different loci on them. And so again, genes that are close to each other, they are going to be more linked, so they will be they'll show up like that. To map distances and to map orientation, what we need to use is what we call a minimum of 3-point test cross. In 3-point test cross, you're looking at 3 different genes, not 2. So you can use that third one to orient which one comes first, which one comes second, which one comes third. The first thing you want to do is identify your parental gametes. Which one are going to be your parental gametes? So for example, in this one, it's going to be little a, big B, C, big A, little b, big C. Those are your parental gametes. Then you need to second classify the recombinants. Once we have the recombinants, we can use the formula from before to determine recombinant gamete frequency. And then we need to add in conditions where there are double crossover, because remember, there are going to be cases, because now there are 2 different genes, 3 total genes, where 2 genes are recombinant and 1 is the parent. So you need to be able to identify those and take those into account as well. Here's an example. Cross results form something like that. So this is a cross where there are 3-point cross. And so you have your phenotypes again, the short or long tail, brown or white fur, and now we are also looking at whiskers, so long or short whiskers. And then you have the number of progeny that are present for each one of them after we did that 3-point cross, so in F2. And then we are looking at the different possibilities of the gametes from trihybrid that would give rise to these particular results, given that your genotype of the F2 cross for each one of these, right? So these are your genotypes. Looking at these genotypes, you know which gametes you were getting, and then those gametes are going to be looked at. And now for each one of the loci pair, so remember, now it's not just 2 genes, but 3 genes. So you have 3 different possible loci pairs. You have AB, AC, and BC. For each one of them, you would look at this particular gamete and say, is it a parental or is it a recombinant? And you would write that in. So here you can see that for the first one, it is a parental between A and B, and then a recombinant for the other 2. So it's a double recombinant. If you have all 3 are the parent, one of the parentals, then that's a parental. If all 3 are, if there's only one recombinant and the other is parental, then it's single recombinant. Okay, so here are your 2 parentals, right? They'll usually have the most progeny with the 2 parental genotype or gamete combinations. And then these are your double recombinants, where you have multiple recombinants, and these are going to be your lowest combinations. Okay, so let's look at our recombination frequencies of the 3-point cross. So first is determine the each pair of loci, as we talked about. Second, we're going to calculate recombinant frequencies, and then we're going to use those to determine gene order or map order. So our 3 loci pairs are AB, AC, and BC. And by looking at that chart behind, we can look at all the progeny that gave AB as the same, and combine those together, and then divide by total number of progeny that we had, which is 120. So I did that for each one of them, and these would be the numbers for each one, and then calculate my recombination frequency. So my recombination frequency came out to be 25%, 10%, and 32%. This is not taking into account double recombinants though, so this would be marked wrong or partially wrong if that was the answer that you would get. However, this is enough to give you the gene map order. So we know that, you know, here BC are the furthest apart from each other because they have the highest recombination frequency, and AC are the closest to each other because they have the lowest. And then AB means B is going to be a little bit further apart. The distance between A and B is going to be more than the distance between B and C, or A and C. So we know the gene order is either BAC or CAB. Oh, sorry, I didn't do it right. Next, we need to also take into account the double recombinants where 2 recombination events happened. So to adjust our map distances, and those are our big A, little b, little c, or little a, little b, little big C, and then correct for those double recombinants. And so we do that because those are double recombinants. We multiply those by 2. There were 2 of them, and you multiply each one of them by 1, and then you recalculate our BC frequency, and you get 35%. So without correction, you would have drawn this map. It could be BAC or CAB, and these are the different centimorgan distances between them. That's basically the same as the recombination frequency, right? However, again, it could be BAC or CAB. It still doesn't tell us which way it went without adding additional genes and then looking at where they are. But what we can tell is that after fixing for our double recombinants, the BA and AC remain the same. They are still 10 centimorgan and 25 centimorgan, but our distance from B to C is 35, not 32 centimorgan. Now, once we have that, then how do we figure out which orientation and how to organize them? To do that, what we will usually do is we will take a segment of our genome or segment of our chromosome, and we would break it up into multiple segments, multiple overlapping segments, and then we use those overlapping segments and do the same combinations, right? We look at all those combinations. We typically do that by taking 100 centimorgan of chromosome at a time. And then here, for example, we have AC, CD, CE, and then so C is in all three of them. Because we have gene C in all three of them, we can use it to overlap these various fragments and to see which one is going to be where. And so we can superimpose these segments, knowing the distances and knowing where they are. So this is the 10 and the B, so we know that's going to overlap. And then look at which way the segment falls to give you all the recombination frequencies correctly. So here, the A to D distance is going to be distance from D to B minus A to B. And similarly, A to E is going to be distance A to D minus D to E. So you kind of superimpose them on top of each other and you get your full map. And in that final map, we know that how each one of those genes are placed, where they are, and what's the distance between each one of them. So here is an example problem for you to try out and try to get the result on which gene comes first, which second, which third, and give me a map order in the comments below using the data that is provided to you. So just to summarize, recombination is defined as any process where your combination of alleles is not the same as one of the parents. The recombination frequency between any two loci is going to be dependent on their position on that particular chromosome. If they are not linked, if they're far away from the same chromosome, or if they're on different chromosomes, it is going to show a maximum of 50% recombination frequency, which follows Mendelian genetics rules. Loci that are very close together on chromosomes are going to be linked, and they are going to give you recombination frequencies lower than that. And so crossovers, which are normal part of meiosis, are what are going to allow for this recombination to occur and give you those recombination frequencies. These recombination frequencies can be used to create gene maps and chromosomal maps and allow us to find out where relative location of genes on each chromosome and then the distance between them. So I hope you guys like this. If you have any questions, write them in the chat and I can explain them in the next video. Take care. Bye-bye.